All right. Okay, good afternoon, everyone. Can everybody hear me? Somebody just unmute and tell me yes or
Yes.
no.
Yes.
Okay, good. I'm glad. So, welcome back, everyone. And we're going to continue our discussion about the function of DNA and how it works inside the cell. Our interest, of course, is particularly in prokaryotes. There are some things which are very peculiar to prokaryotes about the way the DNA functions. But in general, honestly, the principles that we talk about apply throughout all living things. That is how we are able to discuss the central dogma. The DNA is transcribed to messenger RNA, and messenger RNA is translated to protein by the ribosomes. That principle applies throughout all living things. We ended up last time talking briefly about the process of transcription. So I'm just going to repeat some of what I said last time, bears repeating, and maybe add a couple of things to it. First of all, transcription is performed largely by an enzyme complex called RNA polymerase. There it is there. As soon as you see that word, polymerase, you know that a nucleic acid is being made. And in this case, the nucleic acid that is being made is RNA polymerase. You'll remember the DNA polymerases from DNA replication. So RNA polymerase is an enzyme. It's actually an enzyme complex, but its responsibility is to manufacture RNA. In this case, it is manufacturing messenger RNA in the process of transcription. There are other RNA polymerases because there are many kinds of RNA in the cell. And the other kinds of RNA are derived from other areas of the genome. In this case, what we're looking at is transcription of a normal working gene. So we are wanting eventually to end up with some product, probably a polypeptide product from this gene. So what the RNA polymerase does is it initially binds upstream of the gene itself in an area called the promoter. And the process whereby the polymerase finds that promoter is very, very complex and actually poorly understood. We still discover new things about the function, the promoter function. But for our purposes, we're just going to say that the RNA polymerase binds somewhere up here, upstream of the gene, and it then moves down until it receives a signal to begin transcription. That signal itself can be is locked into a set of sequences in the promoter. The RNA polymerase knows how far down to go before it starts producing a messenger RNA strand. So observe again the rule that the RNA polymerase is going to make an RNA strand. This is the new RNA strand that it is making, but it can only add bases at the 3' hydroxyl end there. So what the RNA polymerase does is it opens up a short area of the DNA. It's much smaller than the replication bubble that we saw in DNA replication. This is called the transcription bubble. And the RNA opens it up. And this here is the template strand. The template strand of the DNA is that strand that contains the genetically useful information. This is the actual gene that is being read off. So the RNA polymerase is moving in this direction down the template strand, and it is creating an RNA strand, which for a little while is a hybrid strand between DNA and RNA, but it soon comes off there and the DNA strand rewinds behind the transcription bubble. So there's just a short length of the messenger RNA is kept as the RNA polymerase moves. This is the movement, by the way. This is quite a slow process compared to DNA replication. DNA replication is extremely rapid. It moves thousands of bases a second, but this is a much slower process. So the first thing to remind you of is this template strand here. Only one of the DNA strands for this particular gene. This strand is the template strand, and this is the non-template or non-coding strand of DNA. This, of course, is coding for the template strand. The template strand also codes, not only codes for the messenger RNA, it codes for the non-template. That is how we replicate DNA. Each strand, each strand acts as a template for the other strand. But in this gene here, this strand here contains the gene that is being read off by the RNA polymerase. The second thing to notice, because we're making an RNA strand, not a DNA strand, an RNA strand, is we do not put in thymine to base pair with adenine. Instead, we use uracil. So here, and in this instance here, you see T, it's going to bring in an adenine. Adenine here is bringing in uracil. Here's the uracil already bound, uracil there. Otherwise, GC and CG as normal. The only difference in RNA is that we don't use thymine, we use uracil. So you can immediately recognize an RNA strand by the presence of uracil in the strand. Also notice it is single-stranded. RNA is inherently single-stranded, although we're going to see that some strange things can happen with RNA. It can fold back on itself and base pair its own base pairs. We'll see that in a little while. So this process of transcription will proceed until the RNA polymerase reaches signals which instruct it to leave the DNA strand. The RNA polymerase actually falls off the DNA in response to signals which are built into the sequence of the gene. And when that happens, the RNA polymerase falls off and the transcript, the messenger RNA transcript, is freed. So have a look here and realize that for any gene which is operative, which is being read off, we can have lots of RNA polymerases reading it off at the same time. And this is common for many genes. It's especially marked in bacteria because the messenger RNA actually is free inside the cytoplasm. Something else happens in eukaryotes, but in prokaryotes, this whole process takes place in the cytoplasm. It is not taking place inside a nucleus as it would be in eukaryotes. Otherwise, the rules all apply, complementary base pairing, except U for A, added only at the 3' site, so the strand is made in the 5' to 3' direction of the messenger RNA strand. Eventually, the RNA polymerase falls off and the transcript is freed, and then we have to start thinking about the processes of translation, of what happens to this messenger RNA transcript. Remember what I told you, that the cytoplasmic machinery cannot speak DNA. It can only speak to the messenger RNA. It can only speak RNA. And in fact, what we've got to realize is that here we have an RNA strand. It's a pure nucleic acid. That's all it is. But the product that we want in the end is a polypeptide. It's a chain of amino acids all joined together. Entirely, completely different biomolecule. How is it that we can get from the nucleic acid to a chain of polypeptides? Well, the answer is that we need a molecule which is halfway between the two. If, for example, you can speak English and you can speak German, but you can't speak Swedish. So what do? You go and you find somebody who can speak German and Swedish. You go speak to them in German, they'll be able to just translate everything for you. And you see what I mean? What we need is a translator. We need something that will translate nucleic acid into amino acid. And the best choice is something which has both traits. And this is this fantastic little molecule called transfer RNA. And transfer RNA is a small molecule. It's a small RNA molecule. It's another species of RNA. And it has the ability to bind an amino acid at one end. Okay. Oh, sorry, I forgot. There's these little videos we can watch just to finish off I'll repeat what I've said about translation. Okay, come on. Where are you? Come on. No, why is it not? I can't get it to work. I can't get the animations to come up. Oh well, this is in your PowerPoints. So there are these little animations to help you understand the ins and outs of transcription, and certainly use them for your revisions. Okay, I'm sorry, I can't seem to get it to go. I don't know why. Try again. There. Hold on. Let's do this one at least. Okay, By the way, that's the details of termination you don't need to remember. They were so vaguely put there that I'd rather that we didn't consider it anymore. Just remember that at the termination sequence, there's some signal to the RNA polymerase that it leaves the template strand. Okay, so in order to understand how translation works, we first of all need to look at the organization of the messenger RNA. And the way in which the code works is that in DNA, the bases are organized into triplets, 3 base sequences, 3 base, 3 base, 3 base. And those are translated into messenger RNA into 3 base codons. And the codons stand for something. The codons are a code, literally a code for something. In most cases, they are a code for an amino acid. Other cases, they are a code for an action, as we'll see in a minute. And when we look at just the mathematics of it, you end up with there are 64 possible codons. And 61 of those are called the sense codons. They either code for an amino acid, or in one case, they code for an amino acid and an action. And then the other 3 are codons which are purely instructions, and they're called the stop codons. And they literally do that. They stop translation from occurring. We say that this code is degenerate. It doesn't mean to say it's despicable. It simply means that we only have 20 amino acids that we need to code for. And yet we've got 64 possible combinations of our 4 bases in these codon sequences, in code 3. So there are 64 possibilities for using A, U, C, and G in the codons. But we've only got 20 amino acids. So what happens is that you see that some amino acids, here these are abbreviations for the amino acid names. Leucine, here phenylalanine. But look at leucine, and you'll see the 1, 2, 3, 4, 5, 6 codons, code for leucine. That is why the code is said to be degenerate. Any one of these can code for leucine. So we go through the table of codons here, and we see all the possible codons, most of them coding for an amino acid of one sort or another. And then we see that, first of all, there is one special codon here. And this is one you must remember. I never ask you to remember what the codons stand for. I always give you the table to work from. But this one you must remember, and that is the AUG codon. And that codon codes for the amino acid methionine. In prokaryotes, it actually codes for a type of methionine called formal methionine when it's the first one. So I'll explain a little bit more in a second. So AUG codes for methionine, but that AUG also is an instructive codon, and that is the start codon. The very first AUG in the sequence, after a particular signal, as I'll mention in a minute, the very first AUG is where the ribosome actually begins to translate the messenger RNA strand. So we'll see how this works in a second. But just remember, the first AUG, that is the start codon. But you mustn't forget that methionine is a very, very common amino acid in polypeptides and in proteins. So the start codon is just the first AUG, but the AUG may recur many, many times in the sequence. But so the cell has to have some way of knowing what is the first AUG. and that is given by a signal which lies a little bit upstream. And the first AUG after that signal, that is the start codon. So the other three, remember there are 61 kind of functional amino acid codons. Then there are these three, UAG, UAA, and UGA. These codons are stop codons, and they are an instruction to the ribosome to stop translation. And that is where the polypeptide ends. When as the ribosome runs through the messenger RNA strand, when it reaches one of these, it will stop, and the ribosome will disengage and release the polypeptide. Okay, so here's the way that the table of codons works. First of all, very, very, very important. If you have this in front of you and you have a pen with you, mark here 3'. This side here is the 3' side of the codon. Okay, so it's 3'. It's very important. And if I say to you, okay, I'm going to give you a codon, and I'm going to ask you to tell me what amino acid does that code for. Let me just say CAA. How do you find that? Well, you can chicken peck your way through and find it, but there's a quicker way, and that is I said CAA. The first letter is C. There it is there. So it's somewhere in this block here. Then the second letter I said was A. So we know it's this block, but we know also that it's that block there, because we got C, A, and now I'm going to go A, and there it is there. CAA, glycine. GLN is glycine, the amino acid. So CAA is the amino acid, is the codon for the amino acid glycine. So you can see that if I have a DNA sequence, a template strand, and they are organized into triplets, and each triplet codes for a codon, then RNA polymerase reads off this codon, and eventually the cell has to be able to translate that. We will have on our messenger RNA strand a whole long string of these codons, and somehow or other the cell has to read those codons, and it has to somehow take amino acids from the cytoplasm, and it has to join them together. If I first of all have glycine, CAA, and then the next one I have CGA, arginine, I've got to join arginine to glycine in the cell somehow. And that is the process of translation. And let's just see what they have to say to us.
Professor, is there audio in your side? Because I don't hear any audio on my side.
Sorry, so what did you say? Hold on 1 minute. Sorry, what did you say?
I was asking if there's any audio on your side, because I can't hear the video on my end.
You can't hear can anybody else hear the video?
No.
Oh, shoot. I don't know what to do about that, because I've got the audio and you can hear me. Oh, I'm sorry. Basically, they're saying the same thing as I've just told you. So I don't know what to do about that.
No, I'll try to read the
yes.
bottom.
Okay, all right. Okay. We can do that. Oh, sorry. Oh, it's glutamine, not glycine. Sweet.
Professor,
Yes.
will we need to know the full name of the amino acids, or is it okay to just know the
I would
just what's on
really
the
like
chart?
you. I would really like you to know the names.
Okay.
But I tell you what, I've realized looking at these, what I want to do is actually I'm going to put in another table with the full names in. So
Okay, that's helpful. Thank you.
okay, I think that's it. Sorry, you'll have to try and listen to that on your own to hear the soundtrack, but you basically get the idea.
No worries.
Okay, so let's do a little exercise, and I'm going to ask you what the following codons code for. Okay, so let's just do them in order. CCU. Shout out somebody if you get it.
Proline?
Correct. You're correct. Proline. There it is there. Okay, CCU. There, proline. Okay, AGU.
Serine?
Correct. Again. Okay. There you go. GAG.
Glutamic acid.
Correct. GAA. It's a trick question.
It's the same one.
Correct.
Glutamic acid.
Correct. Correct. And then the last one you already know. And this one you should be able to just tell me straight away. And that is AUG. And as soon as you see that, you know this is methionine.
Can you clarify why GAA is glutamine again? I'm sorry, I didn't.
Sorry, say that again.
Can you clarify why GAA is glutamine?
GAA. There it is there.
Okay, got it.
And it's GAG as well.
Okay, thank you.
Both of those code for the same thing. Because it's okay. So we said that methionine starts the translation process. That is, as soon as the ribosome sees the first AUG, but there are lots of AUGs in that messenger RNA transcript. There is a little signal above where the translation has to start on the messenger RNA. Messenger RNA can contain all sorts of information, quite separate from the codons that it contains. And this is one of the pieces of information. Upstream of the actual gene, of the gene transcript itself, it has a little sequence which is called the Shine-Delgado sequence. And it's actually a place where the one part of the ribosome will bind first. And it will bind to the Shine-Delgado, and then it will move that part of the ribosome to a small portion of the ribosome. Will move down the transcript until it reaches the first AUG. And once that has happened, the other part of the ribosome will come and fit in together, and then they will start the transcript. That's how the cell knows where to begin translation on the transcript, on the messenger RNA transcript. So now we need to talk about this other incredible little molecule, which is the transfer RNA molecule. A transfer RNA, as I said, it's another species of RNA. It's quite a small molecule, and it has two important parts on the molecule. The one is that at the one end, it's a 3' end of the transfer RNA molecule. it can bind an amino acid. Then the rest of the transfer RNA molecule is kind of folded into sort of a T shape, or an L or something like upside down T or an L shape. And at the very bottom, it has a little 3-base sequence, and that is called the anticodon. And that is literally what it is. It's an anticodon. It will base pair with a messenger RNA codon. So in other words, have a look here, and we'll look at a transfer RNA for the amino acid phenylalanine, and it has as its anticodon, it has AAA. Okay, just remember that. Let's just flick back. Let's look up AAA. A. What is AAA? I put the wrong one. I know, I just realized I put the. Okay, it's the. Let me just do this again. I'm sorry, I've got confused now. Yes, sorry, it's the anticodon. It has the anticodon is AAA. So it binds to the codon UUU. Okay, UUU, and there it is there. So our codon for phenylalanine is UUU. The transfer RNA that carries phenylalanine has an anticodon AAA, and you see how it fits there. If this is 3' UUU, the anticodon fits 5' AAA. And you can imagine just picture it here. We've got a tRNA, and on the other end of it here, it's carrying phenylalanine, actual phenylalanine amino acid. Here's our translator. It's a RNA that is a combination of an RNA molecule and an amino acid. So now the nucleic acid part, the RNA, can talk to the messenger RNA. And at the top, it's carrying the translation. It's carrying the actual amino acid that this codon stands for. It's a marvelous system. So it's the same sort of thing. I've given you another one, tRNA for serine. The anticodon is 5' UCA, and that'll bind to a codon 3' AGU. If you want to go and look up, you'll see that one of the codons for serine is AGU. So we're going to watch as this process is used. It means that we have many kinds of transfer RNA. We have many, many kinds of these small translator molecules, because each amino acid has its own transfer RNA, because the amino acid has to be bound by a transfer RNA with the correct anticodon. We can't have methionine bound to a tRNA with that as its anticodon. Each amino acid has its own group of tRNAs with specific anticodons for them that they will bind to. And by the way, the transfer RNA, we find out transfer RNAs get recycled. They pick up amino acids, do what they need to, lose the amino acids, and then they go and get recycled. So all of this magical stuff is performed actually by is performed by the ribosomes in the cytoplasm. Let's just have a look at these. I've just done these to show you. These are the same codons that you saw earlier. And so the 3' CCU protein, and the anticodon is GGA. 3' AGU, serine, 5' UCA, that's the anticodon, and that's the anticodon for serine. So here's the glutamine, it'll be 5' CUC is the anticodon. Here, this is to show you that glutamine can have more than one transfer RNA molecule that will bear it to. And there is an important one. This is the anticodon for methionine, UAC, which binds to the codon AUG, which is both the start and then subsequently will be for methionine in the messenger RNA transcript. Okay, so let's have a look, and we'll go step by step and see how this process of translation is done. And I'm very happy to go over this several times if you need, because there are several steps in here that need to be understood. So here's our messenger RNA transcript, 3 inside the cell. And as soon as messenger RNA appears in the cytoplasm of a prokaryote, ribosomes will try to bind to it. In actual fact, the first binding is by this little unit of the ribosome called, and this is the small ribosomal subunit. And as I told you, it binds upstream here, and it moves to the first AUG after that little sequence called the Shine-Delgado sequence. The ribosomes are absolutely incredible little bodies. They consist of their own RNA called ribosomal RNA, which is structural. Remember that RNA can fold into 3-dimensional shapes. And so part of the ribosome is made up of various RNA molecules and some proteins together. But the enzymatic activity of the ribosomes in translation is performed by the RNA. This is an example of RNA acting as an enzyme. And we say that this activity is ribozymal. It's to remind us this is RNA, but it's acting as an enzyme. The first little subunit binds a little bit upstream, moves down here, and it will check. It will jiggle back and forth to make sure that it's seated exactly right. When it's there, it will allow transfer RNA. Here's the transfer RNA molecule. You can see it's folded into this kind of T shape. And at the bottom here, there is the 3-base anticodon. Well, we have our start codon is AUG. Our anticodon is UAC, and it's going to base pair there, UAC. And look what it's carrying. It's carrying methionine. It's carrying a methionine amino acid. This is just the start. This is initiation of translation. The small unit will bind, check it's in the right place. It will catalyze the binding of UAC here. And if everything is correct, it will allow this large ribosomal unit to bind to the messenger RNA transcript. And this is what we have here. Now, inside the ribosome, there are different, we call them sites. There are places where particular metabolic things happen, as you'll see. The first site here is the P site there. That's where we're going to start off. We start off with the tRNA bound there. There's this anticodon, there's the codon, and there is a rule. And that is that the ribosome cannot stand an empty site here, and this is the A site. And you can think of this as being the amino site, if it will help you remember. This is the polypeptide site. This is the amino site. And there's rule number 1. The ribosome will not tolerate an empty A site. So another tRNA is immediately brought in here. And here, the next codon in the transcript, here we've had AUG. The next codon here is UUA. There it is there, UUA. That is the codon for leucine. And here comes the tRNA with that particular anticodon. The anticodon must read AAU, and that is leucine, for leucine. So that's going to come here, and it's going to sit here. And now now the real meat and bones is going to begin, because the aim of this is to translate these codons. We want to take these amino acids and we want to join them one to the other. And that is performed by the catalytic activity of the ribosome. The ribosome actually forges a polypeptide, a peptide bond to start making the polypeptide. We have methionine here, we have a leucine there, and what it does is, and this is really important, it doesn't take the leucine that way to join it to the methionine. It takes the methionine here and binds it to the leucine. Here's rule number 2. The ribosome will not tolerate an empty tRNA. Okay, so as soon as it sees that empty tRNA, it's going to shift one codon along. It's going to move that empty tRNA into this site here, which is the E site, and is now moved this leucine with the methionine attached. It has moved that into the P site. What's the rule? The rule is that it can't stand an empty A site. It immediately brings in another tRNA, and here it's brought in, it's bringing in CCA, and the anticodon, CCA, matching this codon, GGU, which is for glycine. What happens now? Well, that is going to now bind to that with a peptide bond, catalyzed by the ribosome. This is now empty. The ribosome kicks this out, and it moves one codon along. It's called translocation. It jumps one codon at a time, codon by codon by codon, and it's doing so in response to the binding of these amino acids to one another, leaving the tRNAs empty. They have to be evicted, and in order to fill the site, the ribosome moves along one codon. Here's the next one going to come in, and this is phenylalanine. AAA is the anticodon. What is the codon there? The codon must be UUU for the anticodon to bind to. So, and so on, and so on, and so on. And it can go on for thousands of codons. It can build huge polypeptides. The peptide bond is extremely strong. So these polypeptide chains are very robust, and they actually emerge from the ribosome. There's a tunnel that they emerge from, and they extend out into the cytoplasm, and they can actually start to fold immediately. They emerge. They can start their maturation process. So this continues codon by codon by codon by codon until the ribosome reaches one of the 3 stop codons. Sorry, here's the stop codon here, UGA. There is no corresponding amino acid for the stop codon. Instead, there is a complex that comes in here that actually terminates the process of translation. It is an active process. It requires ATP, but the cell responds to the presence of the stop codon by stopping translation. And when that happens, the ribosome falls off, and the transcript, the polypeptide transcript, is freed, and then goes off to be matured. Okay, so that is the process of translation. But I'm going to do it once more for you, okay? Even if you're bored and understood it all, we're going to do it one more time because it's a complicated process. And so remember the rule, can't have an empty A site, right? If you have an empty A site, then a tRNA has to be brought in to match this. We cannot, the other thing that the ribosome can't stand, it cannot stand to see a naked tRNA. So what is going to happen, this methionine here is going to be bonded, peptide bonded, to the next incoming amino acid, leaving this tRNA without an amino acid. We say that the tRNA is uncharged. So this is if this is bonded to something there, it leaves this tRNA uncharged. And the response of the ribosome is to move one codon down and move that empty tRNA into this site here. It's sometimes called the E site or exit site, but it's not really a site. You see what the sites, the P and the A, are named because they have binding taking place between nucleic acids. There's no binding taking place here at the E site, but sometimes you'll see it referred to as the E site. So that would be the E site. This is the P site, and this is the A site here. If there's an empty tRNA in the P site, the ribosome moves one codon down. If it moves one codon down, it's going to move till it's over there. It moves this one, whatever's here, into the P site. So what here's in this example, we have methionine. It doesn't tolerate an empty A site. Here comes the second tRNA. This one was with leucine. Remember, this is the one key thing to remember, that the methionine now binds to the leucine, not the other way around. You go from whatever is on the P site binds to whatever arrives at the A site. So here, methionine binds to leucine, but that leaves this tRNA uncharged. The ribosome responds by moving one codon down. It moves this empty tRNA into the E site. Now it's got this, which consists of the leucine with the methionine on it now, moves that into the P site. Can't stand an empty A site. It brings in a new tRNA here to match these codons here. Here's the anticodon. This is glycine. It's now going to peptide bind the leucine to the glycine. Again, that leaves this uncharged. As a result, the ribosome is going to move. It's going to kick this out and move this into the E site, leaving this, moving the whole thing, methionine, leucine, and now glycine here, leaving this empty for the next one to come in. And so on, and so on, and so on, step by step by step. The ribosome marches down the messenger RNA transcript until such time as it reaches a stop codon, and could be any one of the three stop codons will code for the same thing. And in response, the cell moves into here a complex of proteins called the releasing factors. And the releasing factors actually terminate the process of translation. The whole thing falls apart, and the whole polypeptide is released to then be taken off and matured by whatever needs to happen. Remember that the polypeptide is only the beginning of the functional protein. Many things may still happen to that polypeptide It may be modified in all sorts of ways before it forms a mature functional protein. So I have, sorry, I'm not going to, I can't show it to you here. The YouTube thing doesn't work when we're using Zoom. But if you want to, there is a little movie here on the process of translation from YouTube, which you can use to clarify the process. And what I do advise you to do is to work with this idea of the anticodon, the codon. Try working out, look at the table of codons, work out what is, for this codon, what is the anticodon? For this codon, what must have been the DNA triplet? If you know the DNA triplet, what is the DNA in the corresponding DNA in the non-transcript side, et cetera, et cetera. Work these things back and forward and back and forward. And it'll soon become even clearer to you how it is that this system actually works. We start off with triplets in DNA. They are transcribed to the codons in messenger RNA. They are translated by the anticodons in tRNA with the catalysis, the enzymatic activity of the ribosome. And the tRNA brings in the correct, appropriate amino acids, all to be joined together to form the polypeptide. It's a marvelous system. Okay, so there's just a couple of things, other things that need to be said about this. First of all, here's one of the differences between the prokaryotes and the eukaryotes. As soon as messenger RNA appears in cytoplasm, ribosomes will start to try to bind to it. In the prokaryotes, of course, there's no nuclear membrane. There's no nucleus. In eukaryotes, this whole process has to take place inside the nucleus. And the messenger RNA is not released straight into the cytoplasm. The messenger RNA doesn't see ribosomes for quite some time in the eukaryotes. But in the prokaryotes, ribosomes can immediately bind to the messenger RNA. Here's this really brilliant electron micrograph. And if you have a look carefully, you can see there, that thin line there, that is the DNA itself. And the DNA is being read by RNA polymerases. And the RNA polymerases are producing a messenger RNA transcript. And the ribosomes of the bacterial cell are immediately binding to the messenger RNA transcript. They kind of, in a way, the ribosomes are almost like labeling where the messenger RNA transcript is here. This is called a polysome. And it's very typical of transcription and of translation in the prokaryotes. Now you can also imagine, you can't see it because they haven't stained for it, but each little dot here is a ribosome, and they are busy reading the messenger RNA transcript, and they will be producing protein from their reading, from their translation of the transcript. So we can have many, many, many transcripts, messenger RNA transcripts produced from the same gene. And on those transcripts, many, many, many ribosomes may read them, may translate that same transcript. So it's a very efficient system. We very soon start to build up a lot of product from this gene, from reading off this gene. So here we have here, and just ask you, here's the DNA, right there. Each of these marks an RNA, messenger RNA transcript. Where's the oldest messenger RNA transcript? Where has RNA polymerase just bound, and where has it been bound for some time? Well, it's been bound for some time where we have the longest messenger RNA. So that's the oldest. The RNA polymerase there has been on for a long time, okay? Now ask yourself, because here's our transcript here, and as the transcript is being produced, so ribosomes are running down it. Which ribosome has been on here for the longest time? That's the trick question.
Is it near the DNA?
Yes, yes, correct. Yes, yes. It's so efficient that the ribosome runs is translating the transcript as it's being produced. So this ribosome here has been sitting there ever since that the very first little bit of transcript appeared in the cytoplasm. And it just sits there as the transcript passes by. It is running along it and it's producing its proteins. We can't see the proteins because they just didn't stain for them. But here in the diagram, you can see more or less how they would be produced. So this is called a polysome. Very, very typical of prokaryote translation. But in eukaryotes, something quite different happens. And in eukaryotes, transcription proceeds much the same way by RNA polymerase, transcribing the DNA, transcribing the gene. And translation, however, has got to take place in the cytoplasm. And so if transcription is taking place in the nucleus, we have to export the messenger RNA from the nucleus out to the cytoplasm for the ribosomes to reach it. In addition, the messenger RNA transcript in prokaryotes emerges mature. It emerges as it's going to be translated. Nothing more has to happen to it. But in eukaryotes, the transcription is quite complex. And in fact, the messenger RNA transcript, a number of things have to happen to it before it can be exported and before it can be read by the ribosomes in the cytoplasm. And the first thing is that the transcript is broken up into sections. Some sections consist of DNA sequence that codes for polypeptide, and those are called the exons. They're, if you want to think of it, that is what the ribosomes are going to read, are the exons. But in between them, there are numerous sections of DNA that do not code for protein, and those are called the introns. They actually are functional. They have many different functions, particularly in signaling, transcription signaling, and all sorts of things. But before the messenger RNA in eukaryotes can be translated, those introns have to be clipped out. And they are cut out, they are edited out inside the nucleus. They're edited out, and all of the exons are joined together to form a mature transcript. And in addition, there are some other things that happen. For instance, a tail of many bases of of adenines is added to the messenger RNA, and there's a little header part that is also added. All signals for the ribosome, and all indicating to the cell various destinations for the messenger RNA. The production of protein in eukaryotes is quite complex. Sometimes it is produced straight into the cytoplasm. Other times, the products of translation are kind of injected into those folds of envelopes of organelle called the endoplasmic reticulum. So there are various locations where translation can take place in eukaryotes, whereas in prokaryotes, it just happens inside in the cytoplasm. So here you can see, here we have exons interrupted by these introns, and the introns are clipped out in quite an elaborate editing, and then the exons are all joined together like that to form the mature transcript. That mature transcript, first of all, I will also add some other things happen to it. It has a tail put on it and a little header region put on it here. But once that messenger RNA is mature, it is exported through the pores, through the nuclear pores in the nuclear membrane. And its destination thereafter often depends on the information contained in this little header region. Oh, you don't need to worry about the method of editing. Just remember that in eukaryotes, the messenger RNA transcript is edited. It has introns cut out and exons all stitched together before it is exported for translation. Okay, so I want to just finish off here. There is quite a lengthy discussion in your text about how genes are actually controlled in the prokaryotes. I don't need you to know all of the details. I just want to point out to you that regulation of gene activity is a very, very complex issue. There are different kinds of genes that we see in operation. First of all, we may have genes which run the whole time, particularly genes which are producing proteins which are used up the whole time or which the cell needs the whole time. Those are housekeeping. They're sometimes called housekeeping genes. They're also called constitutive genes. But many genes remain silent until they are actually needed. Then they are read off, and then they are switched off as soon as they are no longer needed. Very often, as soon as there's sufficient product from them inside the cell, the gene gets switched off. There are many ways in which the cell does this. There are many ways in which it regulates gene activity. But I do want to point this out to you because it's an important concept in prokaryote genetics. And that is, unlike eukaryotes, prokaryotes very often organize their genes in a particular way. And that is that they put a series of genes which are related to one another, one after the other. Very often, these are genes whose products are like enzymes, for example, in a metabolic pathway. So you may have genes for these enzymes that are going to be used sequentially in a metabolic pathway. We actually find them in sequence on the DNA inside the gene itself. So they're put together these are called structural genes. They are functional genes, and they are related to one another in some pathway or something like that. So these are often organized into what is called an operon. And an operon has the following characteristics. First of all, it has a string of these structural genes, related genes. Then it has a little area called the operator. And the operator is like a switch. You'll remember that we said before that one of the, in transcription, the polymerase, the RNA polymerase has to bind to a promoter region. It binds first to the promoter region, and then it moves down to begin transcribing the gene. When we're talking about an operon, not all genes in prokaryotes are organized like this, I'll say first of all, but many of them are. Many related genes are organized like this. They're organized into an operon form. We have a promoter, then we have a switch called the operator there. So RNA polymerase has got to get from there through the operator to begin transcribing the gene. And this is where the control lies. Somewhere remote, often far away on the genome, there is another gene called the regulatory gene. And the regulatory gene is producing a protein. And here it is here. And it's, in this case, it's a repressor protein. It's a protein to switch this operon off. And it's produced the whole time. It's regularly producing this repressor protein. But for example, if we wanted to be producing a product here, what we'll often find is that the final product of a metabolic chain will actually bind to the repressor. So let's say, this is a very famous one. We're producing the amino acid tryptophan. The amino acid tryptophan is produced through a metabolic pathway. It has a repressor protein which is produced by this regulatory gene somewhere far away. When tryptophan appears in sufficient concentration in the cytoplasm of the cell, it will bind to the repressor protein, and that repressor protein now can bind to this operator, and it sits there, occupies that space, and the RNA polymerase cannot move forward. It cannot now read off these genes. We've switched those genes off. As the tryptophan concentration in the cell, but the binding here is not permanent. The binding comes and goes. And so if as the concentration of tryptophan falls inside the cell, the repressor proteins lose their tryptophan, they lose their ability to bind to the operator, and now they open up the operon, and this gene begins to be read again. We start producing whatever it is. This system can also, this is a repressive system. So this is a negative feedback. We've got the product feeding back and activating the repressor protein. But there are ways in which these operators can also act to activate. They can form proteins. They can form proteins which come off in response to precursors. Just to illustrate for you that there are very complicated ways in which the cell regulates whether a gene is switched on or whether it's switched off. Okay, so I think that is probably as much as your brains and my brains can take for the day. And let's finish off. Are there any questions?
I have one question.
Yes, go ahead.
How is the repressor blocked? Like, what blocks it?
It actually physically sits there. When it's out here and it's just by itself, it's unable to bind to this part of the gene. But when it's combined, like I used the example of tryptophan, when tryptophan and the repressor bind together, this repressor can actively bind into the DNA there, and it physically sits there. It physically binds to that part of the DNA, and the RNA polymerase simply can't get past it. It physically blocks it from transcribing. Then as the tryptophan comes away, as concentration in the cell drops, tryptophan comes away, the repressor can't stick there anymore, and it falls off the DNA, and it frees it up for RNA polymerase to transcribe. Does that answer your question?
Yeah, it does. Thank you.
Okay, sure. Anyone else? Hello, I heard someone.
Professor, I was just wondering, it's kind of related to this. So you're saying that the repressor can work either way, though? So there could be feedback where the final product allows the repressor to bind with more affinity.
No, yeah.
Other way around?
There's actually another system.
Okay.
There's another system of activation. Activators work in various ways, but I'll give you an example. The activator sometimes works. It's bound here at the operator and stops the gene from being read off. That's its normal situation. But if a precursor comes in that needs to be processed, the precursor can bind here. And if the precursor binds there, the activator falls off. And then the polymerase can run. I'll give you an example. say, for example, there is a sugar that the cell seldom sees. Well, in order to ferment that sugar, it's got to push it through a whole metabolic pathway. It's no good having that pathway running the whole time if the sugar isn't around. So in that case, the gene would be silenced. It would be silenced permanently until the sugar itself appeared, and the sugar itself or something allied to it binds then to the activator, and the activator falls off, and now all the genes can be read off to process the sugar, to process. So that would be an example of an activation system.
Okay, that makes sense. Thank you. I had one more question about the tRNA, and
Yes.
wondering when with the specific tRNA, how it's holding the amino acid, what end of the tRNA is it bound to? Is it on a 3' or 5' side of the tRNA?
3'. 3'. Yes. It's always that hydroxyl, that 3' hydroxyl that participates in any of these kind of reactions. Yeah, it's bound at the 3' hydroxyl end.
Great, thank you.
I have another question.
Yes.
So before translation happens, you said that the ribosome binds to a different portion of the DNA. So that's
Yes.
always the Shine-Delgarno portion,
Yes.
right?
Of RNA, of the messenger RNA. Yes, the Shine-Delgarno is in the messenger RNA. So here it would be slightly above here. It'd be slightly above there. And the first step, actually, they haven't shown it here, but the first step is that the small ribosomal particle binds at the Shine-Delgarno, and then it moves down till it finds the first AUG. And then it signals the first tRNA to come in, and then the big subunit joins. So a lot depends on this little small ribosomal subunit. It gets the whole process started. So the Shine-Delgarno is somewhere here upstream of where translation will occur, and it moves down to the first AUG that it sees. Then after this, it may see many, many AUGs, but they're not the first. So it just treats them like a normal codon after that. I'm sorry, did that clarify it?
Yeah, thank you.
Okay. All right, everybody. As you go through this, don't forget that tomorrow, 1 o'clock, I'll have a question time for you. Usually I open it up. If nobody comes within the first 15 minutes, I shut down. But you're well, if I can try and come in, at 1 o'clock or so, and it can go on as long as you want. And it's the same meeting ID as last week, but I'll send it out again. I'll post it in announcements on Canvas for you. All right. So everybody, don't forget, please get ready to vote. We have less than a week to go. Go vote early. Carry your ballots if you filled out absentee ballots. Do whatever you can, but this is a very important election. Please go and vote. All right, and I'll see you next Monday.
I have a question.
Yes, go ahead.
Is there any way you can record like you know how you do the meetings on Thursdays at 1?
Yes.
Yeah, is there any way you can record them, because I have class
Sure.
during the time?
Sure. Yes, I can. Very few. There have been some questions asked, but there has not been much participation. So there's not a lot. Not a lot has happened. There have been 1 or 2 issues to cover, but I can certainly do so. Yes.
Okay, thank you.
If a recording doesn't appear, you can assume that
Yeah. Okay. Thank you.
Whatever. That's a pleasure. Okay, everybody. Yep. Ciao. Nice to see you all. See you next week.
Have a
See you
good
next
night.
week. Have a good day.
Thank you.